It seems to work. Okay. Hello again. Thanks to the organizers, first of all, for inviting me to give this talk. Today I will present this work that we developed in the context of the PRECISE project during last year. PRECISE is an EU grant that has the aim to develop computational models and personalized models to understand better prostate cancer. Our main aim in inferring this clonal composition, given multiple biopsies, is that we want to decipher cancer heterogeneity, and we want to do it by finding and detecting proliferative subclones and initiating mutations. This can allow us to develop personalized models and understand better the progression of the disease in a specific patient. Our focus in this work is on whole exome sequencing. You can wonder why we want to work with whole exome sequencing in this problem, but actually we didn't want to go for single-cell sequencing that could allow us to have a perfect resolution for this type of problem, because still there are some challenges implied in single-cell sequencing, and it's an expensive technology. And actually, what happens in the clinics at the moment for very heterogeneous cancer type, like, does it work? Okay. Like prostate, is that usually when a patient came to the hospital and biopsies are collected, we collect different types of biopsies, multiple biopsies. This is done mainly because, as you can see, the tissue is very different. And actually, when we consider subclones that are characterized by specific mutation, if we don't take care of this spatial resolution of the cancer, it can be a problem because we can lose a specific subclone. In this case, here we lose this red, yellow, green subclone in the second biopsy. So actually, looking at multiple biopsies help us a lot, in the sense that when we have whole exome sequencing experiment, basically for all our mutations that we are able to detect, we have an estimation of the frequency. So this is interesting in the sense that we can exploit the fact that we have multiple biopsies to define co-occurring mutation patterns and try to understand, using this mutation profile, how this mutation can be sorted in a phylogenetic structure. In the sense that our main hypothesis here is that mutations that are high in frequencies in multiple biopsies should be at the root of the tree, and then these mutations are inherited by all the children of the initiative subclone of the root. And what is interesting is that the tree structure comes naturally from the partial order induced by the mutation in the different samples. So up to now, and especially when you consider stable genome, the situation is pretty easy to deal with, because when we have a fixed number of allele in every mutated cell at a specific genomic location in every subclone, the mutation that we observe from whole exome sequencing can be a good approximator for the true frequency of mutation. But actually, this is not the case when we have to deal with a stable genome, like in cancer, and especially in some cancer types like prostate or hepatocellular carcinoma or colorectal cancer. So at this point, the frequency that we measure, the observed frequency, is different from the true mutation frequency. So what we want to do is to exploit the spatial information given by the multiple biopsies and accounting for this copy number alteration. Let's start with a simple toy example where we consider a cell population. So we have a bulk data, a population that contains only two types of subclones, two types of cells. Some cells that carry a specific mutation and the other cell that in this genomic location doesn't carry a mutation. So the true mutation frequency, phi, in the biopsy S for the mutation M, the red mutation, in this case is easy, is 0.5. Half of the cell in our population carries this specific mutation. When we look at the average total copy number in the sample, we just do an average between the two different populations. And this total copy number is distributed across the various types of mutated allele that we have, the wild type allele in the mutated cell, the mutated allele in the mutated cell, and the number of copies in the non-mutated cell. Actually, also in this very simple case, we see that the observed frequency is different from the true mutation frequency. So if we want to put all these ingredients together in a system of equation, what we can have is that we can link the total copy number alteration to the weighted sum over the non-mutated cell and the mutated cell of the different types of allele that we have. And moreover, the most important fact is that we can link the observed mutation frequencies and the total copy number to the true frequency and the mutated allele in mutated the copy number of the mutated allele in mutated cell. Actually, when we look at real tumors, the problem is a little bit more complicated because we have to take into account the purity of the sample. So not all the cells that we have in a biopsy are tumor cells. So in order to do that, we have to introduce another variable, this observed copy number. As you can see, in this system of equations, there are many unknowns. But for some of these variables, we can find estimation, for example, for the copy number that we observe, for the purity, but actually there are still too many unknowns. What we propose to solve this problem is an algorithm that is called Chimaera, that is using a 3-steps approach to estimate this mutation frequency and then let us build phylogenetic structure for a specific tumor. The first step of Chimaera is an initial guess. Like we try, okay, let's consider the copy number. When we do an initial assumption that the total copy number, the average total copy number, is equally distributed over the allele in the mutated cell. These are very rough estimate, but actually allows us to define an approximated true mutation frequency for each cell, for each mutation in each biopsy. And these are all known variables in the sense that this is the observed frequency. We can estimate from the reads the copy number using various tools like VarScan, other things, and also the purity can be estimated with different software tools or can be also estimated by clinicians at the moment of taking the biopsy. Once we are equipped with this approximation of the frequency that accounts for the copy number, what we can do is that we can try to find co-occurrence mutation pattern, in the sense that we can try to cluster together mutation with similar approximated frequency and compose our subclones. So what we have to do in this step, what we try to do is that we want to have a robust clustering. Actually, these measurements are characterized by noise, so we want to use methods that are able to deal with outliers and discard mutation that doesn't fit the model so well. And in the 2 versions of Chimaera that we implemented up to now, we used the first version, tclust, is a well-known package. You are using different techniques to define the number of clusters, and in the last version, HDB scan. That has no need to define an estimate for the number of subclones, but actually works directly on the distribution of each subclone, of the mutation of each subclone to try to define the optimal number of clones. The last step of the algorithm is actually the estimation of the frequency. So now we have our group, our subclones, where we have a certain set of mutation for all the biopsies. Actually, this is, again, the second equation that we have seen in the model formulation. These are all observed quantities. We assume that we are able to measure these variables. But still, we have too many unknowns. So at this point, we do two assumptions, 2 hypotheses. One comes naturally from our definition of subclone. So subclones are composed by cells that are genomically identical. So the copy number for a single mutation that belongs to this subclone is constant over the different biopsies because we observe the same subclone. And the second assumption is that given a specific sample, the frequency of the mutation that belong to the subclone are constant in the sense that they are co-occurring. They always occur together. So at this point, we reduce the number of unknowns in this system, and we have to just estimate the true mutation frequency for each biopsy separately and for each mutation in the subclone. So we can try to solve an optimization problem, a nonlinear optimization problem, where we basically solve the problem as a matrix factorization problem, where we try to estimate by minimizing the distance between the outer product of these true mutations and the copy number of the mutation from the real observed data. At this point, when we solve this optimization problem, what we have is an estimation of the true frequency for each subclone, and we are finally able to build our phylogenetic structure using the partial order of the mutations, of the group of mutations coming from our tumor. So in order to validate Chimaera, we tested it on some simulated assays and on a cohort of hepatocellular carcinoma, HCC cancer. So for the simulated assays, we generated different tree topologies with associated frequencies. And we generated the allele copy number by using and estimating the parameter, looking at mutational profile from different cancer types from TCGA. We focused on breast cancer, prostate cancer, and we also used some samples that were supplied by the University Hospital of Zurich in the context of this EU project. And afterwards, we added a uniform noise to the read generated by the different mutations belonging to the subclones. In this way, we generated around 400 datasets, and we benchmarked Chimaera with other methods that try to solve somehow the same problem, inferring the phylogenetic structure given multiple biopsies. So in this synthetic data analysis, we focus mainly on two aspects. So the mean error that we do by putting together mutations that have different frequencies in a subclone, and the number of mutations that we are able to actually assign to subclones. So the results for Ancestry are pretty clear. Ancestry works in a different way from Chimaera. It solves the optimization problem all at once. So here we simply see how the assigned frequencies differs from the true frequency that we use to generate the data. But actually, we immediately understand that it's doing a very poor job in assigning mutations to subclones. We then tested another state-of-the-art method that is called Schism. Here we see that we slightly improve the frequency. Schism works in a similar way to Chimaera. It tries to cluster together concurrent mutation, but actually doesn't account for the copy number. So we see that the variability of the estimate increase, and still we improve the job that we do with mutation, with the percentage of mutation that we assign to the subclones. When we ran Chimaera on the synthetic data, we realized immediately that accounting for copy number in this specific set of data sets helped a lot. And the frequency estimation is improved and the variability is reduced. Still, we have a comparable result under the mutation percentage point of view. And these play really a key role when we look at real tumors, because actually these robust clustering methods put together mutations that are confidently assigned to a subclone. So the problem is that when we run all these three methods on real data, on castration-resistant prostate cancer patients and on HCC cohort, they were not able to produce estimates and include in the phylogeny reconstruction more than 10 to 20% of the mutations. So this is why we tried to implement a new version of Chimaera that is able to preserve more mutation in the synthetic data, and that actually gave very interesting result on real data. Here we see that we pay a price. We estimate a little bit worse the true frequencies. The variability is still okay, I would say, but we gain a lot in terms of mutation preserved. So we are able to consider much more mutation. In the synthetic data, maybe this is not striking, but when working with real tumors, this is very important because we have to be able to use what we have from the data set, basically. For the application on the HCC cohort, we consider this cohort that was recently published in Cancer Research. We have 9 HCC patients, where for each patient we have 5 different biopsies. So when we apply Chimaera on this cohort, here I'm reporting some results put in a toy drawing, where we basically put together we have all the subclones found for these specific patients in all the 5 biopsies. And we see that before the estimates, when we look at the mutations that are clustered together, that are occurring after the frequency approximation, if we display the normal frequency, we see that there's a variability that basically makes it impossible to find a partial order between these different groups of mutation. But actually, when we apply Chimaera estimates, the situation is much clearer, in the sense that we are able to build a phylogeny tree based on the partial order induced by the subclones. We then try to see if so this was basically a personalized approach. So we looked at a single patient, then we tried to look at multiple patients. So we look at which mutation was more frequent. As expected, it was a TP53 mutation. And the interesting thing here is that the TP53 mutation is always assigned in the initiating clone. It's always present with a frequency, but it's also more interesting to notice that this TP53 mutation in this specific cohort and for these patients was always appearing together with a loss of heterozygosity. So considering the copy number here helped a lot to increase the true mutation and assign the TP53 mutation to the subclones. This is very interesting because TP53 has been associated with initiation event and proliferation in HCC. We try to explore deeper the results that we obtained by looking at all the mutations contained in the initiating clones for all the patients that we had, the 9 patients. And what we found is pretty interesting in the sense that when we do a simple pathway enrichment analysis for all these mutations found in initiating clones, we found an enrichment of the Wnt signaling pathway. This is very interesting for 2 reasons. So Wnt is known to be related to initiation and proliferation of the tumor. But actually the second most important aspect is that for HCC, there's a correlation between the signaling pathway and initiation and proliferation of the tumor, but only in presence of a catenin B1 mutation. Catenin B1 mutations are very frequent in HCC. Usually they appear in 20 to 40% of HCC patients, but actually here, as you can see, is not present. So this seems to suggest that Chimaera has a potential also to try to basically find certain type of treatment that are related to Wnt inhibitor. So HCC patients are treated with Wnt inhibitor only in presence of a catenin B1 mutation, but actually from this analysis of the cohort, it seems that there are also other mutations that suggest the activation of this pathway in HCC patients. So we have seen that Chimaera is able to study a single patient, but then by looking at specific cohorts or group of patients, we can also translate and transfer what we learned at the personalized level to the disease level. So Chimaera at the moment is available as a web application. You can ask a login, put your favorites if you have some biopsies somewhere, and try to run Chimaera, look at the estimate, and then you can tell me how it works in your case. And so what now? At the moment, we focused specifically on estimating the true mutation frequencies, but actually what can be really interesting is focusing also on the automatic reconstruction of phylogeny. At the moment, we were lucky in the sense that we always found a partial orders for the samples that we processed, but actually sometimes this partial order can be not so easy to find. So we have two ideas in mind at the moment. One could be plugging in Chimaera estimates inside other methods that build phylogenetic structure, like Ancestry and Schism before, or developing something that can try to find a partial order using, for example, the copy number estimates that we obtain from the Chimaera optimization process. Another interesting thing that we want to do specifically for HCC is that we want to, so what we did is that we had a very small data set with these high quality data, multiple samples for each patient, but actually what we can do is then going back to larger cohorts for this type of cancer and see if what we found with these frequency estimates can be also applied in other cases. This is very interesting. And there is also a plan to apply Chimaera to the prostate cancer outcome cohort from the University Hospital of Zurich in order to do the same analysis for prostate cancer. And we are currently working in the implementation of our REST API for Chimaera. And with this, I would like to thank the European Union for the funding and all the groups involved from the European project. And if you want to ask something or anything, discuss, chat about Prague, tomorrow I will have a poster at the poster session, or just write me an email. Thanks, many thanks.
We have time only for one quick question.
Hi, I'm just wondering, did you try comparing Chimaera to any of the phylogeny methods that integrate copy number aberrations?
So we tried to run, actually I don't remember exactly which method because it was a work that we started last year, but we then decided to only use Ancestry and Schism because other methods were not giving satisfactory results in the sense that we required a lot of manual tuning of the parameters in order to give some estimates on our synthetic data. But we didn't want to somehow overfit the synthetic data because then applying it on real cases, it would have been a little bit weird. There's a plan to try to apply Chimaera and also these methods, including methods accounting for copy number on other simulated datasets, but we weren't able to find datasets where we had this estimate, where we had like frequency, a read of the frequency and an estimation for the copy number as a ground truth. But yes, it's true, we should, I mean, it's still a work in progress. We will surely integrate these methods. But at the moment, at the time we tested them, still there was not so, they were not giving interesting results. But it's surely something that we have to consider. Thanks.
Okay. Thank you again.
Thanks.